Okay, good. This thing is tethered here, huh?
No kidding.
High technology. Okay. Let's see. Okay, we need to put this in display mode. Do you know how to do that? Just click down there in the bottom. That's not it. Okay, I got it. There we go. Okay. Now let me get my mic back on here. Is it time to go for drinks yet?
Oh, it is. It better be good, big guy.
Okay. I know I'm the only thing between you and the reception for our awardees, so I'll try my best to be brief, although if I am, it'll be the first time. So I want to congratulate Deborah. This is a picture of Deborah at a small meeting that we were at together. Almost every time I'm with Deborah, there's alcohol involved, and I found this one picture out of my many, many pictures of Deborah, and where there wasn't, a wine glass or a beer bottle here. But then I remembered, after I put the picture in there, that I think something Royce had in a small bottle had been tipped into that coffee mug. So, afraid I can't do that. Anyway, congratulations, Deborah. I've enjoyed every drink we've had together. And I told Lane, years ago, I said, when he was starting his first job at IU, I said, you got to have a group, philosophy, when you start your own research group. And this was the one he came up with here. And you can see, here he is, with his colleagues exhibiting his group philosophy, which, in science, this is a good one to have. Okay, well, let me get on with my talk here. I've been interested in actually stimulated by George Whiteside's work with easy to use and in resource limited area type sensors. And what my group has been doing for the last few years is developing self-powered microelectrochemical devices. So I'm going to tell you two quick stories that are related to those. So my interest in microfluidics and microsensors started when I did a sabbatical like 12 or 13 years ago at Aclara Biosciences. I was really fascinated by microfluidic devices. What this is showing is a PDMS microchannel. It turns out there's some hydrogels that have been photolithographically immobilized within the microchannel, just a little pad. It doesn't fill up the whole microchannel. And you could, we put cells in there and all kinds of things. And then you could run a fluid over those hydrogels, and things could diffuse in and out of the hydrogels, and they could change color and so forth. But, what became, and the appeal of this is that it's, small. But when you pull back, from this image, what you see is that there are a lot of lines going into this microfluidic device, a lot of tubes. And when you pull back further, you see all this stuff here, a microscope, pumps, and all of this stuff. And so the question is, what can you do to start getting rid of some of that instrumentation that would be, difficult to implement, except maybe in a centralized analysis lab? And so one answer to that are these handheld processors. And the idea here is that you have a little sensor strip, of course, a handheld reader, and you put the sensor strip into the reader. And that's good too, as long as you're in a place where you have a reader and batteries and people that don't drop the reader too often. Okay, but the ideal situation would be these little paper fluidic devices, and that you would just read these out just with your eyeball. And so there'd be nothing more to the analysis than just something, a little piece of paper like this. Okay, so that's the direction that we're going. I wouldn't say we're there yet, but we're heading in that direction. So here are the two stories that I'll tell you. This is the first one. This experiment was done by a student in my group named Brian Zacchio. And what he did was he made a self-powered sensor for trypsin. And the device design is shown here. Schematically, there's a block of agarose, and there's 2 cells carved out of the agarose. The agarose acts as a salt bridge in this experiment. And in one of these wells is placed a protein layer, and I'm going to explain to you what that's for as we go along here. And then just an iron 3 solution in the what we call the cathodic well here. And then if you look at the bottom, this is just a glass slide. It has 2 microfabricated gold wires that act as current collectors. And then there's an LED connecting those 2. And then this is a small piece of magnesium foil. Over the magnesium foil is a small piece of Reynolds wrap. And then there's just a mask to define a particular region of the Reynolds wrap. All this is put together. It's a little ugly when it's all put together, but you can see it here. This is it's about the size of a quarter. Here's the LED that's lit up in this case. This is the agarose block here. Here's this well here. Here's the iron well, and then there's ionic conductance through the agarose. Okay, so that's the idea. The reason we were interested in trypsin is partly because we could work with it, okay, did what we wanted it to do. But also it turns out that at certain concentrations in blood, above 1.25 micrograms per milliliter, people suffering from pancreatitis, have these elevated trypsin levels. Okay, so here's a better representation of what happens in this device. So you remember in one of the wells here, the anodic well, there was a protein layer, and this protein layer is basically whey protein isolate. So it's not so different from this protein powder that you might buy at the grocery store to put in your smoothie. And here's the aluminum layer, the magnesium layer, the gold current collector, the LED, a wire connecting to the other half cell, the iron 3 solution, and then this is this agarose salt bridge. Now, if I have trypsin and I add trypsin to this to the protein layer, trypsin does what trypsin always does, is it starts chewing up that protein layer. And then in a second step, I'll add a little sodium hydroxide. Sodium hydroxide, now that the protein layer is porous from the trypsin, sodium hydroxide will easily penetrate through the protein layer and start dissolving the aluminum. And that now connects the magnesium foil to the solution in this cell. And now the cell is activated. There's a voltage of 2.2 volts between the 2 half cells. And this lights the LED, which has a turn-on voltage of 1.7 volts. Now, if there's a sample added that doesn't contain trypsin or something a lot like trypsin, then and then the development step, which is the addition of sodium hydroxide, is carried out. The sodium hydroxide doesn't penetrate through the protein layer, and so the LED doesn't light. In fact, when sodium hydroxide is added, this is just a representation of this whey protein isolate in this gel layer. When sodium hydroxide is added in the absence of trypsin, what happens is this whey protein isolate is actually cross-linked by the sodium hydroxide and actually makes it the protein layer even less permeable than it was before the sodium hydroxide was added. So the variable here that we look at is the time to turn on the LED sensor. And that's what's shown in this slide. This is the clinically relevant region here for trypsin concentrations from about a half up to maybe, well, you'd be in bad shape if you had this much trypsin in your blood, but it's a little over 1 microgram per milliliter. And then that's plotted here as a function of the time for the LED to illuminate. So you can see as the trypsin concentration, whether it's in buffer or in serum, as it gets smaller and smaller, the average time to turn on becomes longer and longer. And then this is the negative control here, and you can see up here, well below 0.5, the values for the turn-on time for 0.5 micrograms per milliliter of trypsin you can see these turn-on times here are, 100 seconds or 100 minutes or something, on that order. Okay, so here there's basically a battery built into the sensor, and there's a switch, and the switch is represented by the analyte, and the analyte turns the switch on and off, but no external power supply is required. Okay, so that's the first little story, and then the second story. And Tito, I didn't bring my watch, so let me know.
I'll let you know. If
Yeah.
they're gone, it's time.
I thought probably you would. So we're also interested in these, paper-based fluidic devices. And, the first of these, at least that I know about, was introduced by Unipath in 1988. And there's a great recent review of molecular diagnostic devices in this reference here that was recently published in Analytical Chemistry. What happens here is that a drop of solution is added to this lateral flow device here, and there's lots of different arrangements for these, but basically the solution that might be carrying analyte flows along this piece of paper here, and embedded in the paper are probes that can react with the analyte in the drop. When there's a binding event, something happens. For example, gold nanoparticles might become closer together to each other, and there'll be a color change that's usually read out just with your naked eye colorimetrically. Okay? And they have all of these, really wonderful properties. And lots of these have been sold for years and for various things like home pregnancy. Okay, so this was taken to another level by the Whitesides group and over the last several years. And I'm not going to spend a lot of time on this. You read their paper here. But basically, they came up with ways using photoresists or wax printers to define channels in paper. So the paper ranges in width from, this says 70 microns here. We have some that goes up to 250 microns. And so, for example, you can buy a commercial wax printer for $700, and it'll print wax in certain locations on the paper. And then if you heat it up a little bit, the wax goes into the paper. And where there's no wax, it's hydrophilic. Where there is wax, it's hydrophobic. You can add a drop of solution, and the solution will be confined to the paper channel where there's no hydrophobic wax present. And so they've made devices that look like the ones I've shown here. This is a wax device. And you can put a receptor here, here, and here. The blue here is the wax. Okay, add your analyte here. The analyte will come up to this point, split into 3 aliquots, and move into the test zones. And then, again, colorimetrically, you read out the results of this assay for 3 separate analytes. You could see this one was done with photoresist, and you can see it's much higher resolution. So, but it's also more complicated to do the lithography than to do the wax printing. And then this is another device of a 3-dimensional, multi-level micro paper analytical device, again from the Whitesides group. And what they've done here is they've taken pieces of paper and defined channels in them, just as I've shown you with a wax printer photolithographically, and then double-sided tape through which holes have been punched using I think a laser cutter in most cases. And then this tape is sandwiched with different paper levels together to make a 3-dimensional device. And this is just some pictures from their 2008 paper on this, showing the channels and the tape. You can see here. Here's the tape between two layers of the paper. Okay. And then let me talk about electrochemistry a little bit, also pioneered by the Whitesides group. This is an example from one of their papers. It's a little hard to see here, but there are 3 electrodes here. There's a piece of paper that spans the 3 electrodes, working counter and reference electrode, and then a drop of analyte is placed here, and then it moves along the paper, spanning the 3 electrodes, and you can do an experiment. And it's a little hard to see, but this is an actual device from their lab. And then Chuck Henry, who's here at the meeting. I saw him around, and actually my former student, Orwan Chalapukal, have also done electrochemical experiments. And so here you could see, here's the hydrophilic parts of the paper, and they've just used screen printing to define working counter and reference electrodes. And you add a drop of analyte to the center of this device, it splits into 3 aliquots, and then there's some assay here built in, to those 3 electrodes. And you could do 3 simultaneous assays here. And of course, out of the picture is a potentiostat in both of these cases that's hooked up to these electrodes, okay, to measure the current response. So motivated by these types of studies, a student in my group, Hong Lu, who during his formative years in China, while the other kids, around the world were out chasing each other around and hitting balls with their bats or feet or whatever they were hitting balls with, he and his colleagues in China were folding up paper into origami structures. And so he read one of the Whiteside's papers and came up with a variety of ideas for making self-powered and also origami-type microelectrochemical devices. So let me start with the electrochemistry part here first. So what you're looking at, it's a little complicated slide here, but basically this is a paper-based device. It consists of 2 ITO coated glass slides with a piece of paper sandwiched between them. And so it's a little hard to see here, but here's one of the ITO pieces here. There's wax on the paper that defines a channel so that when an analyte eventually is added to the paper here, the analyte will wick into the paper. Okay? These schematics here show you what's happening on the 2 ends of this device. This is the battery end of the device. This is a self-powered microelectrochemical system. Here's the current collectors, the 2 ITO current collectors. Don't worry about the red here, just look at the blue, okay? So there's a carbon, carbon that's been placed, carbon ink that's been placed over the ITO. And then there's a piece of paper, which is a separator, okay, between the anode and the cathode of this little battery. And then there's a piece of metal. In our case, that'll be just another piece of Reynolds wrap. And then another piece of ITO here. So this is an aluminum oxygen battery, basically. And the main thing that's in it is this little 3 millimeter by 3 millimeter piece of Reynolds wrap. And as you'll see, that's enough to drive the device for long enough to do the assay, 3 or 5 minutes. Okay, over here at the other side, this is the sensor side of the electrode. Of course, it also has current collectors, and it also has built into it, in this case, it has glucose oxidase. And so what happens over on this side of the device is if there's glucose present in the analyte, it's converted by glucose oxidase to gluconic acid, and this ferricyanide is reduced to ferrocyanide. The presence of ferrocyanide is detected by this bottom current collector, okay, and it's reconverted back into ferricyanide. That generates an electron, and that electron is pumped via the battery up to the top ITO current collector, where it converts Prussian blue, there's a little Prussian blue spot here, to Prussian white. Okay, so this is an electrochromic readout of the presence of glucose in an analyte. Okay, now the analyte here is glucose in a physiologically meaningful concentration, but not in a physiological meaningful matrix. It's in artificial urine. If you have glucose in your urine, you have problems. Okay. But for proof of concept, that's okay. Now, the other thing is when this device is made, it's made dry. And to activate the device, remember the battery only lasts for a few minutes. So what you do is you pee on the whole device, okay? And the urine activates the battery. The battery is shipped dry, but the urine provides an electrolyte and immediately activates the battery. And the urine also provides the analyte that you're looking for. Okay? This is just the parts that go into the battery. You can see here's the carbon paper, carbon ink over some paper. Here's a paper separator. There's a little piece of aluminum foil here. Just the different parts of the battery. Real simple stuff. Okay, this slide is just showing you the voltammetry of Prussian blue. It's just to show you that Prussian blue has reasonably reversible voltammetry. By controlling the size of that electrochromic readout spot, the Prussian blue spot, you control the sensitivity of the device. Smaller spot means a lower limit of detection. A bigger spot means a larger limit of detection. If you have multiple spots ganged in series, then you can increase the dynamic range of the device. Maybe a small spot first, larger spot, ganged together maybe through 3 intermediate spot sizes. Okay? This just shows you, this is data for the battery. This is the black line is the open circuit voltage over a period of about 5 minutes. You can see it starts going down. And this, the red line is the short circuit current of the battery. Okay? So the battery is operational. Okay, now, you're up, okay? Get the mouse and click on both images. First that one, okay, then that one. Perfect, thank you. Okay, of course you can't see this spot in this light, but if you could, can you see the spot? Okay. From my angle, it's hard to see. This is artificial urine being added to the device with no glucose present and with 10th millimolar glucose present. And this these movies are speeded up by about a factor of 2. Honestly, I'm staring into this Klieg light here, and I can't see a thing. But is it gone yet?
Yes, it's
Spot's gone? Okay, we can move on. Anyway, and this one's still here. I can see that one, that it's still there. So anyway, it works, and then you would just pitch the device. These are the spots, close-ups of those spots. This is with glucose before the analysis. This is after the analysis. You can see the spot goes away. And this is the control experiment just with artificial urine with no glucose present. Tito, where are you?
I'm right here.
How am I doing?
You got about 5 minutes.
5 minutes, okay. All the time in the world. Okay, so what we've done, what I've shown you so far is a paper-based device that really is just like any lateral flow device. The paper is just pretty stupid, and it's just directing the flow of the water. What we want to do is take that system, that microelectrochemical device I just showed you, and put it in a much cheaper all-paper format. So I think probably most of you know that origami is a Japanese word that means folding paper, okay? You can learn about, it's really complicated actually, and you can learn a lot about it from reading books like this, and there are people that spend their whole life studying origami and folding algorithms, okay? And here's how someone spent their time. I just grabbed that off the web, but you can do all kinds of interesting things by origami. Okay, so this is Hong here, and what he's showing you is the beginning of one of his origami sensors. These are these devices are made using wax printing, and he's printed out a paper. These are just office printers, okay? And he's cutting out the origami device with a pair of scissors here, and you can see there's 6 of them on this piece of paper. You can get an idea of the scale. And then here's one that's been folded and then, sort of unfolded, so you can see its 3-dimensional shape. There's no special alignment required for these devices, because when you do the wax printing, you leave a fold between the wax, so it automatically folds where you want it to fold. So the alignment is all taken care of. Okay, so here's one of them, that he's made. This one actually was done with by photolithography, so the features look a little bit nicer. This is the origami device printed on a flat piece of paper. Here it's been folded up. The edges have been clipped out here just so it'll fit into a clamp in a minute. This is the top. This is the bottom of the device. So he's just flipped it over. And here it is in this little stainless steel clamp. And you can see there's 4 ports for adding different samples to the device. So here's a simple assay that we borrowed from one of Whiteside's papers. If you look here on this level of the device, this is level 3 of the device. There's glucose detection reservoirs. There's 4 glucose detection reservoirs. And if you look on level 5, there's 4 protein detection reservoirs. Okay, so what we'll do now is we have these 4 samples. And for example, take a look at sample 4 here. Sample 4 is the green one. And so it will be added to level 1. It'll work its way through these the other layers of the paper. And eventually, if we look at, I've got to get away from these lights because I can't see what I'm pointing at here. Eventually, it'll find its way, for example, over to this location on the bottom level right here, indicating this level turns blue, indicating the presence of protein. Okay? And likewise, sample 4 will end up in these reservoirs here, okay, these glucose detection reservoirs, and they'll turn brown. Okay? So it detects the presence of both glucose and protein. I was going to say, one really good thing about this origami approach is that, of course, the device is folded up, so it's really simple to make. But the other nice thing is detection can be done not just at the bottom of the device, but anywhere in the intermediate folds, because at the end of the analysis, you unfold it and you read the assay. So here, the glucose assay was done on level 3, and the protein assay was done on level 5. Okay? So let me conclude then, because I'm sure I'm out of time. Tito's pacing.
You're good. I'm just printing.
So let me just read some conclusions here. So this idea of building self-powered electrochemical sensors is perhaps a way to begin to think about avoiding readers. Readers provide, I think the main thing they usually provide is power, okay? But they also have pumps in them and sometimes reagents, so all kinds of things. So you have to worry about how you're going to put all of those things onto a piece of paper if you're going to replace them, at least for some rudimentary applications. I've shown you 2 examples for powering these systems. We actually have another one that involves the streaming potential, but I haven't talked about that today. And I will say that in my experience, at least in our hands, most of these assays are pretty qualitative, yes or no type assays. And of course, one of the things that we would like to do is make them much more quantitative. For this battery-powered device that I showed you with the ITO, we'd like to get rid of that ITO, obviously, because that's the main cost of the device. It's no problem. We're working on it. We know how to do it. It's just a matter of fooling around with the experimental parameters. And then we need to start working on some assays that somebody really cares about, and we have ideas for that, and we're beginning to get head in that direction now. So with that, I'll thank Deborah again and Lane on the occasion of their awards. Thank you for listening to my talk. Where's the bar?
First SEAC meeting and then there's
Oh, sorry.
First
Okay.
first. Any questions here?
One of the things the readers bring, yes, they bring power and stuff like that, but I think the major thing that they bring is the ability to connect individual results to a national net.
Yeah.
For epidemiology and global health surveillance. And
Sure.
health surveillance and things like that.
Yeah.
How do you see this connecting to that, which I understand is a sort of a sine qua non for doing that sort of medicine?
No, that's a real good question. You know, I think, first of all, that there are many applications where somebody needs to know something that it doesn't necessarily need to be connected into a network. And it may just be what's in my drinking water that I'm going to, put in my mouth right now. And, it would be great if that could be, assayed, for a whole continent, but maybe it isn't necessary. And, speaking as someone who's never been to, really these resource, limited areas, I'm guessing that there are still cell phones there. Yeah, and so I think the answer is, these cell phones, at least the smart ones, seem to have everything imaginably built into them. You can put an iPhone under your pillow now, and it'll tell you, they have really sensitive accelerometers in them, and it'll tell you if you have sleep apnea, okay, with an app that you can download from the web. So I think probably at least colorimetric and maybe even fluorescent assays could be done that way. You know, I have one of these, my wife gave me one of these little LED lights. They're like these super bright lights that have a little battery, and you could shine that. I mean, that gets away from the idea of not using power, right?
Even your cell phone has one.
Even yourself, mine doesn't, but I'm sure yours does. Yeah. Good question.
Any more questions?
Do you need some amplification strategies to lower the detection limit? I mean, you have some ideas about that?
Yeah, sure. You know, it just depends. You know, what you need to do when you do sensor stuff, I think, is probably most people know, is you need to start with what you're sensing and, you know, what the level is you're trying to get at. And there are some things that have pretty high levels, like that trypsin sensor I showed you, and other things that are there, you know, at the level of, you know, 6, you know, molecules or targets per, you know, 10 microliters of blood. So it depends what you're trying to sense. But there are, you know, for DNA and things like that, there are all kinds of amplification strategies, you know, coming out of, you know, I'm going to use the same word again, but it's a completely different field, DNA origami. You know, DNA can be amplified actually pretty easily using catalytic DNA. So there's ways to do things like that. Your usual enzyme amplification that electrochemists are. Mm-hmm. Yeah, I don't think it's a problem. Yeah. Mm-hmm. Deborah, last word.
So we're going to be strewing, in essence, bio-waste all over African paper.
These have wax in them, Deborah. You can light up your.
Okay, so you just
burn up start your cook fire or whatever. Yeah. 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 Right. Of course, this gets away from the idea of no power, but except for that.
Alrighty, folks. With that, let me thank all the speakers for great